So today's guest will help us understand a functional aspect of what could be described as the most enigmatic thing in the known universe, which would be the human brain. Now, it seems like every time we think we've figured out some solid truth of the functionalities of the brain, nature tends to throw us a curveball and sends us back to the drawing board. Now, not to get too philosophical, but you can't appreciate the complexity of the human brain until you understand memory, thought, and consciousness. Now, if you don't believe me, go talk to AI researchers. They'll tell you how seemingly impossible that is. So I'm sure it'll take us a lifetime to really decipher all the neurological processes of the human brain, but we don't have that kind of time here today on um, conversations on science, right, doctor? So that's why we're just going to narrow it down to, one aspect, which would be working memory, short-term memory, right? So I'd like to welcome Dr. Shaw. Dr. Shaw is a neuroscience researcher at the prestigious Rockefeller University in New York, and she's a member of the Rajesethupathy Lab headed by Dr. Priya Rajesethupathy. Now, in her bio, it says here she's a recipient of the Women and Science Fellowship in 2017 and was awarded a grant from the Robertson Therapeutic Development Fund, which supports the translation of her research to serve clinical innovations and the public good. So I'd say welcome, doctor. How are you doing today?
I'm good. Thank you. Thank you for having me.
All right. So, your qualifications kind of speak for themselves, right? But I still want to hear it from you, right? So, you're originally from Taiwan, right? And somehow you've made your way to the United States. You did your undergrad in the United States. You did your graduate studies as well. So, I'd like you to kind of walk us through your research journey up to where you are today at Rockefeller University. So, and by the way, why Rockefeller University?
Yeah, Rockefeller University is located in New York, New York City. And I've been always, since I'm from Taiwan, I'm always seeing New York City as like a symbol of like American dream. So
Mhm.
that's where I choose to do my postdoc, my PhD and postdoc studies. So when I grew up in Taiwan, I grew up in a very um, like industrial thing. When you think of Taiwan, you think about like semiconductor industries. So my first thought is I'm going to build some kind of biomedical devices. So I joined biomedical engineering department in University of Rochester, upstate New York. And there I got exposed to bioengineering. And that's how, and I remember reading a paper coming out from one of my fellow Taiwanese, Dr. Li Hui Cai. She's currently a chair professor in MIT, and her studies study an enzyme called CDK5, an enzyme that is specifically uh, um, present in the brain and has a function of building synapses. So synapses where neurons and neurons talk to each other. Um, so I'm like fascinated by this little proteins that have such important functions and can like even if you disrupt these proteins, animal have very bad performance, cognitive performance. So that's kind of my entryway into the neuroscience. So after I graduated from Rockefeller University with University of Rochester with my master degree, I joined neuroscience department in Mount Sinai, And there I was able to really study how synapses are built. We study a developmental disorder, autism disorders, and and so that exposed me to um, how important it is of the gene of the human genome. So autism is like other psychiatric disorder, have some genetic component. You can inherit those genetic mutation from your parents, and then the product of this of the mutant gene disrupt some kind of neural connectivities in the brain. So so that's how I get interested in how this blueprint of our brain um, can changes the way we our can changes our behavior. Um, and so that's
Wow. Cool, cool. So that's a very in-depth explanation of your career. So I know we're going to talk quite a bit about short-term memory, which would be working memory. So by the way, is short-term memory the only kind of working memory?
Um, there are divided by different kinds of short-term memory. Your short-term memory can have visual short-term memory, and you can that is, and you also have short-term memory that's related to speaking, like the words, and you have short-term memory about your motor functions, how you move. So the most studied one is probably the visual system short-term memories. There are tested. There has been because we can do the similar test in the monkeys to have the saccade. So monkey was trained to um, remember a stimulus from different locations on the screen, and then they remove the stimulus, but the representation of that stimulus still present in the monkey brain. So monkey then receive a signal, and they can move their eyes to where the signal was before, and then they got reward. So monkey got really good at it.
Mm.
So when they put electrode in the monkey's prefrontal cortex, they see a signal that is persistent during the delay, even though there is a not signal on the screen. So that's where the the biological substrate, neurobiology substrate of working memory was began to be understood.
Interesting. So if we want to maintain and update task relevant information, so we're using our working memory, right, to get the job done. So is it always working memory? Is it always short term in nature, right? So working memory, is it always short term or could it be long term as well?
Uh, Working memory is defined as most likely the short term.
Mhm. Okay. So a properly functioning working memory, I guess, remembers what is just witnessed, right? While also being able to anticipate incoming stimuli, right? So that's basically what you're saying.
Yeah. So that's more of the different pathways. Remembering the visual stimulus. So there are three pillars. Well, there are many components that constitute working memory. First, you have the sensory input, and then there's a maintenance of that representation in your brain. And then there is a manipulation and execution part. So manipulation is when you are in different situation, different contexts, you respond to that stimulus differently. For example, if you are in the classroom and you hear a ringtone, your response is probably tune it down. But if you are walking outside and you got a call, you probably pick it up and speak differently. So that is a manipulation part in the working memory. And then there's the execution part. So after your brain decided how you're going to respond to that stimulus, then then you have to execute to respond to that stimulus. So so it was being said like working memory is the mother of all the cognitions because it just has so many components meshed into this process.
Interesting. All right. So what you're saying is with working memory, right? We're able to actually tune certain stimuli away and depending on, I guess, the circumstances, right?
Mhm. Yes, depending on the circumstances. So it
Okay.
has it's a very important component in learning also. Mm.
All right. So now, before we get too much into the weeds, right? So let's go into your lab, right? So you're trying to carry out an experiment to figure out how memory works, right? So walk us through this process, right? What are you doing exactly?
Yeah, so because like it is just described, there's so many components in the working memories.
Hmm.
So it's really hard to Um, so we have to be really focused. And so we designed a very simple test. So we think the working memory has a genetic component in it. So the brain was built by the neurons, and neurons built with proteins, which is your block, and proteins are built from the genome. So different genetic factors can influence our behavior performance. And a lot of neurogenetic scientists has shown this before. For example, there's fly mutations in the flies that was done by early neurogeneticist Dr. Simon Benzer. He isolated mutant flies that have problem learning. So you can pair a shark with a particular smell, and then the fly approach to the smell, it got food shock, and remember, don't approach that smell again. So there's some part of the mutant fly that doesn't learn that. And that's how he isolated a fly gene that, the mutant, the mutation make the flies cannot make that association. So we are doing the similar approach. So we have a pool of genetic diverse animal mice. So those mice are come from all different sources. There are wild derived mice. There are five that laboratory mouse strains that is inbred for many generations in the laboratories. And then through a very complicated mating scheme, we have a population of uh, 200 mice, and they have the hugely variable genetic constitutions. And because of the genetic contributions, so the diversity is so huge, their behavior variability is also very big. So what we did was we have all those 200 mice perform a working memory test. So mice have innate ability to want to go into explore the new places. So we designed a maze that has 3 arms, and the mice run in this 3-arm maze for 5 minutes. So we count how many arms that they enter sequentially, but not overlapping, so they don't come back to the arms that just come from. Um, some mice that perform really well, they always entering a new arm that they never explored before. And some mice keep entering the same arm. And then we can score them um, um, so we can find out their the genetic variation and their behavior variation. And that's how we figure out um, which part of the genome is responsible for the changes in their behavior.
Interesting. So before we get into genomics, right? So let's try to understand the physiology of the brain itself. So, to the best of your knowledge, can you segment the brain for us and its components that actually have to do with memory?
Yeah, it has to do with the traditionally, it has the working memory function has been linked to the prefrontal cortex. So prefrontal cortex is this part, the frontal part of the brain, and also represent the front part of the mouse brain. Um, since I use mouse as a model, so I'm more familiar, I just trying to explain from the mouse point of view. Um, so it's been shown that if you disrupt for example, your lesion, the prefrontal cortex in the mice, they fail to perform the memory-related task. Um, so um, however, recently there are new studies that shows other parts of the brain, for example, thalamus, which is in the middle of the brain that connecting the sensory input from the lower body and also transmit your functions to move your limb from the brain to your limb. So it's kind of the bridge sitting the front and the back and also your spine. It's like the middle of the brain, and that the thalamus is recently being implicated in modulating the function of prefrontal cortex.
Interesting. So, according to one second. So, according to one of your papers, there's a phenomenon that is thought to correlate with working memory, right? So, this would be a constant firing of the cortical neurons. Now, do you suspect that this is the brain's, I guess, flow state when it comes to working memory?
Yeah, so it has to be something that is changed in the brain in the activities of some neurons in the brain as a substrate of memory. And it was thought that there's persistent activities in the neurons in the prefrontal cortex is the most correlated to the working memory. But then there are questions like, why is like this particular neurons has this persistent activity, but not other neurons? Is it because like some repressions of other neurons to receive some inhibition so they don't fire like this persistent activities? Or is there are some like input from other part of the brain, like outside of prefrontal cortex, that tells these neurons, well, hey, keep firing. So, we are interested in all these hypotheses. We take a different approach. Instead of sticking electrode into many parts of the brain, we use genetics. So the idea is, well, if we find these genetic factors that has something to do to tell these neurons keep firing and like memory these things. Then depend on the gene product of this genetic factors, we can probably locate it other factors that manipulate this neurobiology, this neurophysiological phenomena. And that's what we set out to do using um, a um, um, large scale phenotyping to find the link between the genetic variation and the behavior variation.
Perfect, perfect. So let's go back into your lab, right? So you walked us through it, but let's do that again. I like that mental experiment we just did. So walk us through an experiment, all right? So a good amount of physiological experimentation is comparative, right? So meaning we use animal models. So in your case, like you said, you use a lot of mice models. So can you walk us again through the genetic traits we account for before these, I guess, working memory experiments are carried out?
Yeah, so because so we use this mouse population that is um, genetically diverse. The reason they are genetically diverse is because they are crossbreeding of the different laboratory inbred strain and the outside derived, the externally derived, the wild derived mouse strain. So, um, so what we call in the genetics, the strain is they have a stable genetic representation. In each strains are different. They have different genetic markers in their genomes that we can, it's called a single nucleotide polymorphism that we can use as a marker to tell like, well, this one is different from that one genetically. So when we cross them, you got one type of genotype from their father and the other from their mother. So therefore, you increase the genetic complexity. And when you do that for many, many times, you can imagine their offspring will be mosaic of their parental phenotype, parental genotypes. Um, so this cohort, this population of 200 genetic diverse mice have the equal representation of the founder strands. Um, and that's how and that's how that's how they can have a huge behavior diversity.
Interesting, interesting. So we are looking for a diverse population, right? So I guess you don't want that to pollute your experiments if I guess the genes are just too similar, right? Is that what you're saying?
Yes,
Okay.
yes. So because they have many genetic markers, the very high density of genetic markers that can help us to do a very fine resolution genome mapping. Because um, if you want to, if you finally find a segment of your genome that is associated with the behavioral variance, it is important how big that segment is, because if you have a huge segment, there will be like hundreds of genes in that segment. How do you know which one is more causatically related to the different behavior? So the smaller the mapping, the higher the mapping resolution, the smaller the segment you can associate it to your behavior, the easier for the downstream analysis. So you have very few genes in that segment, and that's how you can maybe do some perturbation experiment to narrow down which one. So you have fewer candidates, and so you can figure out which one is more causatic to the differences you see in the behavior.
Interesting.
Yeah.
All right, interesting. So let's see if we can connect this to human anatomy, right? So the whole point of doing comparative medicine is to um, you know, translate knowledge from our comparative experiments to human beings, right? So in your paper, chromosome five comes up a lot, right? Now, what is it, and is there an analogous, I guess, chromosome that is, I guess, possessed by primates and by extension human beings?
Yeah, so that's interesting. So the genetic structure, which by that I means different genes where they are located on the chromosome, is not very conserved between mouse and human. However, each individual genes they do conserve. They are for example um, um, a lot of genes, if you um, express those mouse version of that genes, they will make a mouse version of that proteins, and it can replace the functions in the human cells, the human versions of it. So that's how, by that I mean like it's conserved. So the structure of the genome is not conserved, but the function of the proteins are conserved.
Interesting. Interesting. All right. So is chromosome 5 where the G protein I guess coupled receptor, so the GPR12, is that where it's located? And we know that this is what functionally promotes working memory, right? Am I correct on that?
Yeah.
Okay. So yeah, so chromosome 5, right? That's where that G protein is located.
Mhm.
All right. So in humans, the GPR12 is a class of GPCR, which would be G protein coupled receptor. And it's related to the cannabinoid receptor family. Is that correct?
Yeah.
All
It's
right.
structurally very similar.
Very similar. Now, I'd be remiss not to ask you this, but what does cannabidiol, so CBD, does it actually have any like true effects on memory? So it's all the craze nowadays, right? Everyone is using CBD. So what do you know about this, about the effects of CBD on memory?
Yeah. So I don't study the cannabinoid signaling. I will leave that to the experts in the cannabinoid field. However, um, uh, CBD, first of all, cannabinoid receptors responds to um, uh, the THC. So THC is a agonist that activates the cannabinoid receptor. However, cannabidiol, which is the CBD, is not an agonist of endocannabinoid receptor. It's more uh, and it's more as a antagonist. It is compete with the THC, which is the agonist. Yeah. So, um, uh, and the second point, this is the G-carboprotein receptor 12, although it's structurally similar to endocannabinoid receptor, um, it's not activated by THC. So we don't, it's an orphan carboprotein receptor, which we call it orphan because we don't know what's the ligand. We don't know what can activate it. So it's still further research has to be done to understand what is the endogenous, like what is the natural activator of this receptor.
Interesting. Interesting. So, as a huge fan of CRISPR technology, what role did CRISPR play in your experiments? Right. Anytime I hear CRISPR, I'm always excited to hear what a scientist is using it for. Right. There's so many functional use cases, right? So that's why the creators, Emmanuel Charpentier and Jennifer Doudna, was so quick to receive a Nobel Prize in chemistry, right? But yeah, so how are you using this miracle technique?
Yeah, I use this as a tools to validate one of the to understand more about the functions of this G-coupled receptor GPR12 that we identified because it's not well studied. So um, our experiment will, let's just say, hey, how about we remove this GPR12 from the genome and see how the cells responds. So we did a simple experiment. We have a wild type cells that contains GPR12 and a mutant cell, which we use the CRISPR to knock out. So by that, I mean remove a part of the GPR12. So there's no more GPR12 receptors in the cells. And then we apply glutamate, which is a neuroexcitatory agent. So after in the normal neuronal cells, when you apply glutamate, the glutamate bind to the glutamate receptor, and then neural will have changes in their membrane potential, and they fire. So they have a more active calcium response. And we found when we remove the G-coupled receptor, their calcium response is decreased. They have fewer response to a glutamate. So this has something to do. So it tells us whatever GPR12 does, it has something to do with modulating this calcium signals inside of the cell.
Wow, interesting. Interesting. So are you using Cas9 or Cas13 to actually do the edits?
Yeah, we use Cas9. Um, what it does was it break double-strand DNA, and then cells has its self DNA repairing process and to ligate the DNA back together. However, a little part was removed. And that part is very important to make GPR12 from the genes to mRNA to the protein. So they are missing the beginning part of the GPR12 gene, and therefore the protein cannot be made, and therefore there's no GPR12 proteins in the cells.
Oh, okay. Wow. So that makes a lot of sense. So if we were to round this all up, would it be safe to say that the thalamus, the prefrontal cortex, the hippocampus, are the three segments of the brain responsible for working memory? But I guess the billion-dollar question would be, how do these different segments actually communicate, right? That's where I guess the witchcraft lies, right?
Mhm. Yeah. So um, the all the sensory signals, like for example, your touch and your more or less go through the thalamus and then to input into different part of the cortex. The visual signals also go through the visual part of the thalamus, which is called LGN, and then where the and then go to the visual part of the cortex. Um, and so to answer those questions, our hypothesis is somehow thalamus sending out a signal to instruct the prefrontal cortex um, to maintain um, the particular stimulus to maintain that the persistent activities. And without the instruction of thalamus, the prefrontal cortex cannot maintain or the signal, the persistent activities decrease gradually. And then you remember things for shorter time.
Wow, interesting. So I guess we can segue into a much more practical, much more applied way of looking at your work, right? So neurodegenerative diseases, right? So what role does that GPR12 receptor play in neurodegenerative diseases? And in relation, I guess, to receptors, how can we mitigate these um, deleterious effects, these morbidities, I guess?
Yeah, so when we find out this GPR12 gene is associated with working memory, we're very excited because working memory decline is a signature or is a big symptoms of like Alzheimer's disease. They have working memory decline. Uh, but other disease, for example, schizophrenia, they also have working memory issues. They also have working memory declines. Um, so there is no genetic data linking GPR12 in the human to the Alzheimer's disease. However, GPR12 does, there are human data, genomic data linking the mutations in GPR12 to how the response to the psychiatric medications. We are this line of research need more effort. Yeah,
Okay.
GPR12 and the human um, uh, uh, brain disorders.
Okay. So what you're saying is there's still room to do more investigation on how these things truly work, right?
In the human
In
brain.
the human brain, at least. So let's switch gears a bit, right? So I'd like to touch on a common theme in your career. Now, you've worked in a few labs headed by female scientists. Now, has this been by design? Because I, for one, I really appreciate the diverse views female scientists bring to their respective fields, right? So I don't know if you've noticed, most of my interviews, I tend to alternate them, right? So I'll have a male researcher on, then I'll have a female researcher on, and so on and so on. So,
Mm.
is this something that you actually uh, are cognizant about when you're actually picking a lab?
Um, it's actually happened by coincidence, but I do really appreciate my mentors, my PhD mentor, Dr. Deanna Benson. She she's she's so different. We I interestingly they have different management styles coming to the lab. So then Dr. Benson is very hands-off, and it could also be because I was a PhD student at the time, so there's no like uh, time. There's no like, oh, you have to finish this experiment within this month or this week. So that gives me a lot of rooms to try and figure out which experiment works, which experiment doesn't work. However, after I got my PhD and got into postdoc, then they start to feel the pressure because it's a more like a transitional period. And in the scientific careers, it like I always wanted to become independent researchers. So this like having been mentored into to perform to do experiment, um, uh, it's very different from a PhD student.
Interesting. So let's see. So you did your PhD at Rockefeller as well, right? Or Icahn
I did my PhD at Mount Sinai, Icahn School of Medicine.
Icahn School of Medicine. Actually, so take us back to Icahn School of Medicine. So you started with synapses out there, right? So the smallest neural computational unit. So a young aspiring scientist listening to this right now, would you advise them to take the same steps that you took? So start small, right? So find a manageable task.
Mhm.
And from there on, at least have some sort of grand vision, but start small, right? Is that how you did it? Or am I just reading too much into, what I
Yeah,
read in your
that's
statement?
exactly how I did it. So it's more of because synapse is very interesting structure. Synapse is where neurons and neurons contact and where the information is passed down from one to the other. Um, and synapse is like what you said, is a small, less computational unit in the brain. So that give me uh, um, a more reductionism kind of point of view, like okay, so you see this behavior and you know this neurons is activate during this behavior, but what instructed? What is upstream? What is the downstream? And then it's come down to the synapse. And as you know, many neurodevelopmental disorder like autisms um, is being described as a synapse function malfunction because mutations in autism tend to produce a mutant protein that changes the function of synapse. Um, so that's how I approach the very complex phenomena. I look first into the small part, the synapse transductions. Um, but yeah.
Okay, cool, cool. So what about lab size, right? So I don't think there's a definitive answer about whether being a graduate student in a big or small lab really helps your academic progression, but what is your take? I'm interested to hear what you think.
Yeah, so um, I've been staying on the smaller size in my PhD and in my postdoc. So I can tell the benefit of staying in this small lab that you have closer relationship to your colleagues and more time to your PI to your boss.
Yeah.
Um, and that's a benefit of smaller lab.
Okay, interesting. All right. So you're more of a fan of a smaller lab than a big one.
Yeah.
All right. So what would you say is next for you in your career, right? So I know you did talk about wanting to have your own lab. Now, are you thinking of going into industry, teaching, startup maybe, or you're having just too much fun at Rockefeller currently and you're not even thinking about that?
Yeah. So uh, there is so my scientific interest is still in how brain stores and retrieve memories in different time scales. Um, and there is a part of the nerve systems that is still not very well understood. So there are several cell types, neurons. So neurons, we call them neurons, but if you can categorize them, neurons are inhibitory neurons and excitatory neurons, but you can further divide them into inhibitory neurons like neurons that regulate the local neuronal actions or inhibitory neurons that modulate the different layers of the brain, like more broader but not local, inhibit neurons that regulate other inhibitory neurons. So
Mm,
different types. And then for the excitatory neurons, even more. So you have the excitatory neurons that use glutamate as a neurotransmitters. You have excitatory neurons that use other neurotransmitters as as their ways to talk to the other neurons. So, but there are other cell types, the glial cells. The glial cells are two different major types. One is astrocyte, and the other one is a gliacyte. And the function of those glial cells is just begin to be understood, and they have close relationship to the neurons, especially the astrocyte. Astrocyte have binding the have supply the energy to the neurons, and also we talked about synapse before. Many synapses has been captured by astrocyte. So how does the astrocyte um, uh, affect the neuronal functions, neural circuit, especially the computations? I think that would be the next frontier. Well, it's a lot of lab has working on this, but I think my future research will be focusing on how astrocyte interact with neural circuit.
Cool, cool. So, but you didn't answer one part of the question though. Where are you going to be doing that work? What do you project?
So that's very interesting questions.
Right.
I personally don't care
Mm,
where I work in the industry or in the academia. I'm more like, I can do whatever um, research that interests me. So I don't really says, oh, I have to do it in universities or like in industry.
okay. Okay. That's a very diplomatic answer. I'll take it. So, Dr. Shaw, is there anything else you think we didn't touch on today? I think you gave us a lot of information on how memory works, and I really appreciate it. So, what
Yeah,
do you think?
so although I focus on the discussion of thalamus and its relationship with the prefrontal cortex that has effect on the working memory, I don't exclude that other part of the brain that might have functions in the working memories. And also, from my research, we only touch a very small part of working memory, which more related to the maintenance part of the working memory. So there are other like executions and manipulation part of the working memory that we still don't know what is the genetic factors that affect those or what part of the brain. So there's a lot there waiting for a lot of questions there waiting to be answered.
Wow, I like that. So, if anything, you young scientists listening to this, there's still a lot of work to be done, and you can pick up where established researchers like Dr. Shaw have left off, right? So, Dr. Shaw, I really appreciate this. This was fun. I knew it was going to be great. But if anyone wants to, I guess, read your papers, see what you're up to, how can they get in contact with you, or how can they read about your work?
Yeah, I will send out my informations. You can send informations to my email, khsiao@rockefeller.edu. And my paper is published in Cell. So just Google thalamic uh, orphan G-GABA receptor in the Cell webpage, and you can find my paper.
Perfect, perfect. And yes, she does answer to her emails, guys. So, on that note, Dr. Shaw, thank you. I really appreciate this.
Thank
Bye
you so
now.
much for having me. Bye.
Bye.